So, good evening, everyone. So I'm Loïc Paulevé. I'm very glad to be here. So I'm visiting your lab since Monday. We're here with Dr. Celia Vian, and we're in very good hands with Professor Lee and all his lab. And so I will present you some research we're doing in my group. Where we start from biological system and we want to end up in formal models. So it's quite a long journey. And so my plan was to show you kind of the main decision we took, how we approach this difficult problem. And then I will make a focus on some kind of method that may be of interest of you related to combinatorial optimization and all the keywords that have been presented, which is more oriented towards logic programming. So we are doing a lot of logic programming, and so I will show you precisely what it means. Before we go to So just a few words where we come from. So we come from Bordeaux, which is on the west coast of France. So quite a few kilometers from you, and a few hours of plane as well. So Bordeaux is not a very large city for France, ninth largest city of France. And we're famous for vineyards and the oceans and the nice weather. And we also have a lot of oysters and nice landscape. And cannoli, which is a nice little sweet that we love very much. So we are in a good place to make research. And my lab, so it's like an institute for you. It's a computer science lab of Bordeaux. This is the main institute where we do research in computer science in the southwest of France. And so we have many different kinds of research linked to computer science, from signal analysis to combinatorics, data science, software engineering, and formal methods, where I'm essentially doing my research. And we also have this transversal research project related to artificial intelligence, which is linked to different departments, and also digital health, where we also combine models, data, and image analysis. And for myself, so I'm a senior researcher at CNRS. So I'm trying to organize this research around digital health at the lab, and also at the national level. So I animate a research group with all teams in France related to application of computer science in biology. So as Professor Lee said, I have background in formal computer science. So even though there is biology in the title, don't worry much, we won't do much biology. Just a bit to get the motivation. But it's essentially methodology, computer science, math, all good things. So going to some motivation of what we are doing. So it comes from biology. So you know, when we are created in the belly of mom, we start from one cell, which we call the stem cell, and this stem cell is divides, and when the cell divides, it somehow replicates itself. It's a clone of itself, and progressively, while the cell divides and divides, they will acquire some functionality. Some cell will become blood, some will become bone, brain, neurons, and this is what we call cellular differentiation. So you have stem cells that can, over time, specialize, take some function. And once a cell has decided to be part of the skin, for instance, it cannot change its mind, and it cannot become blood. So it's like we commit to some kind of function, and once we have committed to a kind of function, we keep it. So this is a very general process in biology that we observe at different scale. For instance, for the generation of your immune system that you need to fight the disease. This is a very important process which is generated from kind of blood cells. That so you have a stock of stem cells in your blood that can differentiate and become different type of immune cells to fight different type of disease. So it's a very important process, a very adaptive process. So once cells can also reprogram themselves in the immune system, so they can change of function. And this is also a process we can see in development of diseases, like in cancer, where you have like a healthy cell that we have a normal way to divide and die. And progressively it will occur some mutations and some other changes in its composition that will make it change the way it behaves and will like make divide too much, for instance. So we can also see it as kind of a differentiation where a cell can progressively change of behavior and change of function. So the challenge in biology is to understand this process. So we have a few ideas what is important, but it's something still very difficult to grasp. And it's necessary to understand this mechanism if you want to design new drugs and if you want to cure new diseases, for instance. So just a little of biology. So in the modeling we do, we follow the biology view on this problem. We will focus on relationship between, or the influences between the genes within the cell. So in this drawing in yellow, this is a scheme of a cell, and you have the genome, DNA inside of it, and part of it we call it the genes. And the genes can be copied into a new molecule that have been very famous due to vaccine of COVID, is RNAs. And RNAs can then be translated into proteins. Proteins are molecules which float within your cell. And depending on the shape of the protein and the amount of these proteins in the cell will have different functions. So the amount of protein somehow determines how the cell will behave. So how these proteins are produced is very important, and they are produced by genes between being transcribed. An important mechanism is that some proteins will affect this production. So, for instance, the protein orange may enhance or accelerate the production of protein blue. So you have influences between the gene productions. It's not just straightforward production. You have a lot of regulation. Another important mechanism is signaling. So signaling is a way the cell can sense its environment. So you have like receptors on the membrane, and when you have some particular kind of molecules floating around, it will trigger the receptor and trigger a cascade of chemical reaction that will, in the end, shut down or accelerate the transcription of the gene so that the cell can adapt to a change of its environment and make, for instance, a lot of production of some proteins to adapt. So, what we like to build are models, computational models, that are able to reproduce the behavior of the cell. So the idea is we have the cell that can have different states, so stem state or acquiring some kind of functionality that can change over time. And the idea is to have a program or model that can mimic this behavior. So you will have a correspondence between the state of your program and the state of the cell. And the idea is what we like to do is if the cell is able to change of state over time, we'd like that our program also reproduces this change of state. So you will have like transition and trajectories that mimic what happens in reality. Once we have this model, what you like to do then is you can make experiments within the computer. You can put your model into conditions that you are not able to do in the lab or will be too expensive, and you can see what will happen and try to predict what will happen. And you can also try to think at how can I modify the system such that I change its behavior. So let's say that this corresponds to a cellular state which is bad. We start to divide too much. And we'd like to prevent going here and like to enforce going to this healthy state. So we may try to modify the program to do that, so to change, to drive the system from this state to this state. And we could do that by modifying part of the program. And the idea is that this modification, then, you can then try to implement them in the lab and see if indeed your cells are controlled by your perturbations. So this kind of perturbation in biology, and it's often mutation, gene mutation. This is something we can implement in the lab. We can remove some genes or force some genes to be really active. This is what drugs do indirectly. And so this is how we could predict new drugs, new combination of drugs. So what would be the perturbation to do to your cell so that you force it to go to a healthy state, for instance. And the idea is to be able to predict this from mathematical models. So the challenge is how do we build this model? That's our main challenge. I will, as I said, I will explain to you what's the difference, the path we took, the scientific path we took to address this problem. And I will then focus on a specific part where this is really how we generate this model. So the type of program we use, so this here, now we need to decide a framework. So should it be a C++ program or should it be a Markov chain? So we employ a model that has been used in biology since several years, decades even, which are Boolean networks. So Boolean network is a formal model. It's very simple, and I will explain to you briefly how it looks like a Boolean network. We'll play a bit with a Boolean network, and then we try to think if it's a good framework or not. So the idea is that you have several, let's say, genes or variables that have influences between them. And so the idea is that you will say, okay, the variable is Boolean, it's binary, it's either on and or off, active, inactive, 0 or 1. So gene 1 is a variable, which is either 0 or 1, gene 2, gene 3. And the idea is that you will have rules to decide how, when, in which cases it becomes active, in which cases it becomes inactive. So for instance, you could say that gene 1 will become active when G2 is inactive. Okay? So it takes the negation of G2 and it gives you the state of G. So formally, a Boolean network is just a simple function which maps the binary vectors of a fixed dimension. So you have n components in the system, and for each of them, you have a Boolean function which, given the state of the system, tells you toward which state the component is evolving. So I have another, maybe it's a bit too low here, but you have another example here of Boolean network in this formalism, where you have 3 components, and so for each component you have a Boolean function, so which takes a binary vector and returns a Boolean, and which express how we can compute the next value, the next state of the component 1, for instance. So we take the negation of 2. For the component 2, we take the conjunction between 1 and 3. So 2 become 1, 1 and 3 need to be 1. So it's just for now, it's just a simple mathematical function, discrete function. So if you apply it to a vector, so here the white is for 0 and the blue is for 1. If you apply this function in this particular example, so 1 is 0 and 2 and 3 are 1. If you apply it, you have as a result the vector where f1 is 0. Okay, because You take the negation of 2, 2 is 1, so you become 0, which was already the case. F2 of x is the conjunction between x1 and x3, and as x1 is 0 here, it will be 0. And for x3, you take the same value as 1, so it is 0, so you see that if you compare the two vectors, you see that two components are changing of value. And this is where you can start to think at it as a dynamical system. It means, okay, from this configuration, 2 and 3 can change of state. They are not happy with their current state, they should change according to the model. And you have different ways to model this. Either you say everyone change of state at the same time. So it's like a synchronous update. Everyone updates at the same time, so we call it also the parallel approach. Just you apply F, and it gives you the new state of the system. So this is what we call the synchronous transition. So from this state, from this configuration, you will go to this configuration where everyone is. You could also say, okay, but actually it's maybe not everyone is changing at the same time. Maybe it's first gene 2 that will change of states. And then you are becoming into an asynchronous transition system, where from this configuration you have two choices. Either you update the second component first and you go to this configuration, or you update the third component first and you go to this configuration. So we start now to see transitions, and you see that there's different ways to model the transition. So it's a pure modeling choice, and there are several others. So you repeat this process, and for instance, if you start from a configuration where everyone is 0, so you compute what are the possible next configuration, and in this example, we end up with this possible behaviors. So if you start from here, maybe you will make some oscillation for some points, and then at some point you will converge to one of the two states here. So what can we see from this dynamics? An important feature are like the endpoints. So you see that I say I started here, I do some stuff, and at some point I converge. So this convergence to some states, some configuration, is very important because this is where your computation is going. And you see that you may have several configuration where you can converge to. Not only one. Here we have two different endings. So usually when we model biological system, we want this attractor to reflect stable phenotype of the cell. So when the cell has chosen a function, for instance, we think of it as an attractor. It's an attractor. It's stable. An attractor doesn't have to be a single state. It could be also a set of states in which you oscillate forever. But once you're an attractor, you never leave an attractor. Only your endpoint. You have done your computation, you're here, and you will never leave this attractor. In terms of graph, this is the bottom strongly connected component of the graph. Okay. Over important properties we look for in this dynamical system are the existence of trajectories that link two configurations. It may be important to know that if you start from here, you can reach this configuration and you can reach this configuration. There exists a trajectory going from here. So just the existence of path in your graph. And also the absence of trajectory is very important because this model is a commitment process. Once you have chosen a path, okay, I will go become a bone, you cannot change your mind and become blood. So it means that once you have in certain state, in certain configuration, there is no longer trajectories to other attractors, for instance. Yeah, so as I said, in terms of graphs, all these properties translate very well. But you will have noticed that this graph is exponential in the number of components. Okay, you have 2 to the n configurations. When we do a biological system, we have thousands of genes. So maybe we don't want to build a graph of 2 to the 1,000 states. Regarding the reprogramming that I've briefly told you about before, so it could be that we have these two attractors. We are in the red, the bad one, and we want to jump into the green one. So how can we? So we're stuck here. We want to go here. How can we do that? Actually, in this example, what you could do is that you can block the second component to be always 1. What will happen? So if you are here, you block the second component to be 1. So you will jump here. And once you're here, because you are blocking the second component to be 1, you can no longer take this path. So you have no other choice but going into the green state. So you see, by just changing one part of the specification of our model, very simple modification, I change a function to a constant value, I completely change the attractors I can reach. And yeah, so it could be, for instance, an idea of a drug. If you're on the system, it will be to find a drug that will activate this gene for a while. And so you will drive the system from the red to the green. So to compute this perturbation, then we have different algorithms to do that, either working on the graphs, but also on more symbolic structures. Okay, so that's what is a Boolean network. So you see, it's quite a simple object. So there's been a lot of theory on that, a lot of nice work around combinatorics and dynamical systems. Let's go back to our initial problem. We want to reproduce what the biology is doing, so we can ask ourselves, is this framework a good framework for biology? We may say, okay, this is super simple. Biology is very complex. Just drop this kind of stuff. It's just theory. So there's several points of view of this problem. First, I will just discuss is from a modeling point of view. So one way to see this Boolean networks is actually a fundamental model of a complex system, where what is important is the interaction between components. So the quantity is not important. What is very important in the Boolean network is the fact that this component has an influence on this other component. And so the logic, just the existence of relationship, it's what generates the complexity of these models. And we may think that indeed in biology that may be the case, that the complexity of biology emerges from the fact that you have thousands of interacting components with very specific ways they regulate each other, and that could be actually what is really interesting, not the precise quantities of each molecules. Life is quite robust, okay? If you change a little bit the amount of that molecule in the cell, or you change a little bit the speed of some reactions, you won't destroy the system. Life is very robust. So the quantities are not that important. If you speak to biology, they often speak in a qualitative way. This gene activates this other genes. They don't say this gene at this concentration is very critical. And also, so it's more an argument, let's say, authoritative arguments to say, okay, but in practice they work. There are several studies that employ Boolean network in life sciences, have been able to predict perturbation, implemented in the lab, in the perturbation worked, and they've been able to learn new things in biology. Okay, so that's really modeling point of view. As formal scientists, that's maybe not very convincing to us, so we can rephrase the question. The question could be, are Boolean networks a good abstraction of a quantitative system? So biology is a cell is a quantitative system. You have a number of molecules. Are Boolean networks a good abstraction of it? And this we can think at theorems of abstraction and simulations between different kinds of modeling. So notably, with a Boolean network, can I reproduce any trajectory of a quantitative system? So if you give me a quantitative system, can I design a Boolean network that captures everything the system can do? That's the first question we can ask. Is it expressive enough? The second question we can ask is more something we usually ask in abstract interpretation fields in computer science, is if I make a proof at the Boolean level. So I prove, for instance, that the trajectory is impossible in my model. Can I use that proof to reason on the underlying quantitative system? Can I backport some proofs? Can I reason on abstract level and have results on the concrete level? So when we look at this question several years ago, we actually found an easy counterexample that showed that no, Boolean networks are not good abstraction of quantitative systems. So we were quite sad, but actually we found a way to fix it. So we found a way to make Boolean networks good abstraction of the quantitative system on the formal point of view. So we have theorems now that guarantee that. And so the trick, what we had to invent is a new way to compute the next configurations, how we compute the dynamics. The synchronous asynchronous is not good. You need more things. I won't detail this because I want to speak more precisely over aspects of the work, but just think of it as another way to draw transitions for which we know as a guarantee that if I will move this in another place if I can. No. Yes. Where we have this kind of theorem. So that's my transitions in my Boolean network as shown before. We have several theorems. So the first one is to say, okay, if a quantitative system which is compatible with your Boolean network, it means like you keep the same interactions, but you just add the information on the speed, on the concentration, so you have a more concrete, more quantitative model. If this quantitative model is able to make some trajectory, then for sure you are able to see, you will see it at the Boolean network level. So you cannot lose trajectory. This is something which was wrong with the synchronous or asynchronous. If you just synchronous or asynchronous, you will there's some trajectory you cannot compute. With this most permissive scheme that we created, you have this property. Everything the quantitative system can do, you can do it at the Boolean level. We also have some kind of minimality theorem which says if we do some transitions at the Boolean level, there exists a concrete system that realizes these transitions. So there is no like spurious transition in this Boolean network. And another thing you can derive from that is that if you prove that there is no trajectory linking two configurations at the Boolean level, you have a proof that no quantitative system is able to do it. Because if one quantitative system was able to do it, you will have this like the theorem. So, and this is a very strong theorem, because it means that now we can reject model at the Boolean level. Let's say that you observed in your real system that you can go from here to here. And you have this candidate model, Boolean model. You can already say, okay, this model is wrong. This is not a good model of my system because at the Boolean level, I cannot make the trajectory. So I know that there is no model, quantitative model, that can do that. So it means we can now reject model. So it's very important for the rest of the talk because as we want to find a good Boolean model for the system, we need to be able to say that this model is wrong, this model is good. And we'd like to be able to say it at the Boolean level while having confidence that it will also hold if I have a more precise framework. Another point is complexity. So complexity is super important, especially for practical approaches like this where we try to design models. So as shown you some properties we are interested in studying on these dynamical models. And so the question is, I give you a Boolean network and give you my question, how hard it is to decide the property. So here are the three most elementary properties we look for on this type of model. So the first one is the question, so I give you this function and I ask you, is there a fixed point? So is there a configuration where I have no transition? It's fixed points, it's a single configuration from which you will no longer be able to evolve. So this is an NP-complete problem. It's quite directly related to set. Then you have the reachability. So I give you two configurations and give you F, not the graph. Okay, if I give you the graph, it will be easy. I just give you F and ask you, is there a path, a trajectory from X to Y? So if you do a classical synchronous, asynchronous dynamical system, this is a p-space complete problems, which means very hard to solve in practice. So you need, in practice, all these solvers of p-space, it's exponential space, exponential time. If you do it with most permissive, it becomes a polynomial time algorithm. So it's a huge drop in complexity. Now I ask the question, I give you a state and I ask you, is it a stable state? So does it belong to an attractor? Can I escape from this state? Again, in general, on asynchronous system, dynamical system, it's a p-space complete problem. And with most permissive, it is co-NP complete problem. So not only with this most permissive update mode, you have more guarantees, formal guarantees, but it's also drastically more tractable. You go from exponential time and space algorithm to polynomial time. And in practice, so we did a lot of benchmark, and if you want to do models, so it's what we call model checking, so to verify some properties on a model. Typically, we are limited to 100 of variables with these asynchronous schemes, and with this most permissive, we can go to 100,000 variables without much troubles. Yeah, so this results of QNP completeness. This is something we completed with Young and Professor Lee during our collaboration. So I will come back to this complexity result later, but this has really strong impact on the tractability of the method I will show. Okay, so now I go back to the initial question. I have my cell that I can observe somehow, and I want to build a mathematical model that reproduces the behavior of the cell. So this, what we want to do is a general problem of synthesis. So you have some kind of specification that you derive from your observation. So I observe that I can go from that state to that state. I cannot go from that state to that state. This state is stable. What will be a program that can create it, that has this property? So this is what we call the synthesis problem. You also have this problem in I think it's implemented in Excel. If you give 2 columns of values, like name and first name and last name, you give some examples, and then you can ask, okay, give me the function which from the first name give me the last name. And you have a function in Excel that try to infer the rules to build the name from the last name from the first name. So this is synthesis, so you want to create a model from a specification. So in our case, what will be the specification? So again, I don't want to enter into much details because there's a lot of work here, but we have observation of the system, so it means that from some cellular states, we have like binary specification of the cellular state. Sometimes we don't know if the gene is active or not, so it can be partial, but we may know that from this configuration where some genes are active and some other genes are inactive, later I observe that my cell has been able to reach a state where some state has changed, and I can reach some state where the cell is stable and which has another state. So we have like this kind of partial specification of the graph I've shown you here. So we have existence of trajectory that we observed, state of the genes in these different states, the configurations, and sometimes we can also derive absence of trajectory, so we are sure that there is no path from that configuration to that other configuration. So all the properties I've shown you, we can extract, infer them from the data, from the observations. So this is a data analysis, statistical analysis we do. And we end up with a set of specification that will say, okay, a good model should have this specification. So now the problem is, given this set of properties, can we find the Boolean functions, the Boolean network that have this property? So what's more precise? So if you look precisely what we have to do by designing a Boolean function. So I want to design the Boolean function for this gene, for instance, and I know it may depend on all these genes. It means I have a lot of choices. I can do whatever I want. Either I can say I depend only on B, and I just take the value, or I just take the negation of C. Another possibility is to make the conjunction between gene B and not gene C. So you may think that all the possible Boolean functions between all these variables, and there's a lot. So if you have no constraint on the sign you use in your function, you have 2 to the 2 to the n functions. If you have some constraint on the sign, so it means the function will be monotone. This is still highly combinatorial, so this is called the Dedekind number, and which is also roughly doubly exponential. So that's a huge space to look for functions. And if you fix the function of gene A, it has an impact on the function you will choose for gene B. Everything is intertwined. So if you have 100 functions to find for 100 different genes, everything depends on each other. So you have a huge space and everything is intertwined. And we have no law. So this is really, in biology, we are not in physics, where we have nice E equal MC2, a lot of nice law to guide us to build models. We have nothing, we just have to try and test. So how we tackle the problem? We tackle it as a satisfiability problem. So we transform this problem into a logical problem, and we employ logic programming. So this is what I will detail a bit to you. Maybe you're not too familiar with logic programming. So the basic of logic programming is a SAT problem. So SAT is really related to ILP, so I know most of you know ILP. And this is, in practice, quite relevant to do SAT problem when your variables have a very small domain, like Boolean domain. If you have a large domain for your variable, forget about SAT. And do LP. So what is a SAT problem? So just to refresh your background on this. So the idea is you have a set of variables, of Boolean variables, and then you have clauses. So clauses is a disjunction between variables, so x1 or x2 or not x3. And you have several clauses. And the question is there an assignment of my variables such that all the clauses are true? Okay, so let's try to solve this small problem here. So if I set x1 to 1, this clause is verified. Good. I go to the second clause. x1 is equal to 1, so this is equal to 0. So I have to put x2 to 1. Otherwise, this clause is not satisfied. Okay, so now I have x1 equal 1, x2 equal 1. Good, these two clauses are not satisfied. I go to the third one. x1, not x1, so it is 0. Not x2, it is 0. So I have no choice that not x3 must be true. So x3 must be 0. Okay, so I have 1, 1, 0 as an assignment. Let's try the fourth clause. We're doomed. It doesn't work. So the fourth clause doesn't work. So it's not a valid assignment. So I made the wrong choices. So I need to try another one. So this is an infamous NP-complete problem. So you know that in our current state of knowledge, we have no efficient algorithm to solve this. We have somehow no choice but try all possibilities. This is, you can do that in ILP, just a very simple syntactic transformation of these clauses, right? But SAT solvers use very, very different resolution algorithms. So it's very, very specific way to reason on how to find a good assignment that depend on ILP. So that's SAT. But we are not doing SAT. We are doing what we call answer-set programming. So which still like this declarative programming approach, where you do not program your algorithm, but you program what you want, and then you have a general algorithm which solves the problem for you. So it's very similar in the same approach on SAT and ILP. Things that it has kind of more advanced input language. So you have the syntax, much more advanced syntax you can give to the solver. Yes, some nice subtleties. So I will try to show you a bit of these subtleties. so maybe you will learn something. So this is a very simple ASP program with 3 lines. You can send them to an ASP solver and it will give you some answer. So what do we have? We have facts. So facts are variables which are true. A is true. This is a fact. You have no choice. A has to be true. Then this is a rule which says if A is true, then B must be true. It's kind of implication. And the same here. If A and C are true, then D must be true. The thing is, what are possible solutions to this problem? In ASP, there is only one solution to this, which says A and B are true. So A is true, you have no choice. If A is true, B must be true, so you have no choice either. And then the last rule is also satisfied because C is false, so the implication is satisfied. But in ASP, we will never consider the solution, which is a solution in SAT of this problem, where A, B, C, and D are true. The reason is someone is true in this solution if it is either a fact or it is justified by a rule. So if you put C as true, okay, none of the implications are violated, but C is not justified. You have no way to justify why C is true. So you will never consider in this program that C is true. So this is what we call stable semantics in logic programming. And so this filters a lot of solutions of the SAT problem with this implicit assumption that everything which is true has to be justified. Then we have also choices, so you can have a choice. Either you will consider that none of them is true as a beginning, or just A, or just B, or A and B, so all possible subsets. And in these cases, you have 3 different solutions. So if you decide that nobody is true, then all the rules are satisfied. So this is a good solution. Everyone, nobody is true. Just B is also good. And if you select A, you have no choice to have B. So you need to have A. Another very important aspect of ASP, which really comes from a long work of research in logic. So in France, we really like this kind of fancy logics. And here it's what we call non-monotone logic. So the idea is that in SAT and ILP, the more constraints you add, the more clauses you add, the less solution you have. Right? So the more constraint you add, the less solution. You reduce the space of solution. You filter the space of solution. So this is a monotone behavior. The more rules you add, the less solution you have. This is monotone. In ASP, this is not the case. In ASP, you can reduce the set of solutions. At some point, you will add a rule which will expand it again. So this is non-monotone. And so this is, I mean, even this was motivated by quite philosophical aspects. How can we formalize some philosophical reasoning? And in ASP, it is implemented with kind of a special negation that we call the negation by default. So let's make some simple example here. I'll try to make it intuitive. So there's a character in my story, which is a cat. And the cat was at my place on the morning, and the cat was at your place in the afternoon. And on the morning, I had 100 grams of chocolate, and in the afternoon, I had 0 grams of chocolate. So that's facts, things we collect during the day. And then, okay, I try to investigate why I lost some chocolate in the process. And so I start by making a rule, which is a general rule in society and many societies, which says if there is some character and we have not been able to prove that it is guilty, you're innocent. You're innocent until proven guilty. Okay? So this is what this not means. It's not the negation, the classical negation. It means if you have not been able to prove that this fact is true, then you can apply the rule. So if you are not proven that A is guilty, and A is a character, then A is innocent. So if I add this rule, what you will deduce is that the cat is innocent. And that's the only thing you can deduce. Now you add a new rule. You say, okay, if someone was at my place in the morning and there was plenty of chocolate, and in the afternoon there is no longer chocolate, it means that that character here is guilty, is the one who ate my chocolate. So you have these new rules, and now, well, you derive that the cat is guilty. So this rule no longer applies, and so you created new models that weren't there before. So now you will deduce that the cat is guilty and is no longer innocent. So you create new models. So this is this kind of non-monotone reasoning that uses this not special negation to reason on the absence of proof of satisfiability of an atom. So you see that from a modeling point of view, this is quite this can be very big. So no, I tell you, just you have to believe me, but this is very handy to model problems. We really need very few rules to express what we think, because often we make this reasoning. So it's quite a natural reasoning, actually. Yeah. So this is, for instance, a traveling salesperson problem. I'm sure you have seen it during your studies. So this is, you want to sell something, you need to visit all the cities. So all the cities are connected. So you start in blue. You have a distance between each city, and the problem is in which order should I visit the cities, such that I sell all my things in the minimum number of kilometers. So this is a very classical NP, also complete problem in some sense. How do we model it in ISP? Well, we will declare the facts. So we start in some city. There are 4 cities. Then I declare the roads and the length of the roads. So for going from A to B, I need 10 kilometers and so on. Then I need to choose on which roads I will travel. So for each road, I have a choice, either I take it or not. So that's my choice I have to do. And then I create rules. So I say, okay, so if I start in X and I go from X to Y, it means I visited the city Y. And if I visited the city X and I go from X to Y, I visited the city Y. So it's like you traverse your graph and you call all the nodes that you visited. So this is the rules to generate this graph exploration. And then you add some constraints. You must visit a city. So if there exists a city and you have not been able to visit it, so could be after several applications of the rule, you have not been able to derive that the city is wrong. It's not a good model, so we should reject it. So this is the way we write it. If this is satisfied, the model is rejected. And also, we should never depart twice from the city, from the same city. We should never arrive from 2 different roads to the same city to make sure we just visit each city only once. So that's my constraints. And we can also do optimization. So all at ASP solvers, many of them implement nice combinatorial optimization algorithms. And then I can say, okay, and then minimize the total distance. So you minimize the sums of the distance. So that's a full ASP program. You can give it to a solver and we'll give you the optimal solution of the TSP problem. So you see it can be quite short, and we can include a lot of different reasonings in it. So how do we use that for synthesizing Boolean functions? So the idea is that we want that solutions of our program are Boolean functions. Okay, and that will satisfy some property. So just to give you a concrete idea how we do it, the idea is that we will represent Boolean function in disjunctive normal form, so clauses, but inside the clauses it's conjunction, and outside is disjunction. So this is a Boolean function in disjunctive normal form. You have 2 clauses, and then you can represent it by this fact that for the function A, in the first clauses, you have the variable C, which is positive. In the function of A, in the second clauses, you have the variable A, which is negative. And in the function of A, in the second clauses, you have the variable B, which is positive. Okay, so I just described this DNF using variables, Boolean variables. And how do we choose a Boolean function? Well, we can consider all possible clauses, all possible way to place my variable in all my clauses. So I have a huge choice to make, where for the function A, for the clauses 1 to the maximum number of clauses possible, I can assign some variable to some sign if I can depend on this variable and this sign. So this will generate, consider all possible DNF, and then you add your constraints. Like you may want to be a fixed point, to have a certain fixed point. So you may want to declare that 0, 1, 1 is a fixed point of the function. So you will declare this configuration. So this configuration I named X. So in this configuration X, A is negative, B is positive, C is positive. And then you have rules to evaluate the function, and you say, okay, if you evaluate the function A on X and it gives you some result B, and it is not the same value in the configuration, it means X is not a fixed point, so you should reject the model. So like that, you can write all the properties you want in ASP, and in the end, it will give you only the Boolean function that respects the properties you have written in ASP. So for the different properties we are interested, we essentially have 3 types of satisfiability problems. We have problems of this form. There exists some function, for instance, that satisfies some properties. So this is the most simplest problem we have. So it's MP problems, typically related to reachability and fixed point properties. We also have problems with a second level of quantifier, with universal quantifier, so 2 QBF problems, especially for having properties on the universal properties on fixed point, for instance. So here you can either use bi-level integral linear programming. I haven't mentioned it because I cannot tell everything about ASP, but in ASP it has the same expressivity as 2QBF. So we can directly encode a problem with exist for all in ASP. There is no need to join solvers. The expressivity power of ASP is exactly this class of complexity. And then we have a last category of problems, which is quite difficult because we have 3 levels of quantifiers. And now, even using ASP, we need to combine different solvers to solve this type of problem. Luckily, we haven't identified any relevant problem for us that use 4 levels of quantifiers. Maybe it will happen someday. But now this is kind of the toughest kind of problem we have, and we are actively working on it. So of course, the size of the formula, the size of the number of clauses and number of variables is key in this satisfiability problem, and this is directly related to the complexity results I've mentioned to you. So if you have a linear problem, you have a linear number of variables and rules to write, and it's good. If you have an exponential problem, you are in trouble. So just to conclude on this, so what we are doing with this is that we are building a generic tool to synthesize Boolean networks from properties. So we combine data we acquire in the lab, on which we do data analysis, and also we merge with expertise of the biologists to tell us how to interpret the data. So this generates constraints, Boolean constraints, that we use to generate possible models that can satisfy the constraint and so reproduce what you observe. And from these models, we then have many different algorithms to compute predictions on what are the important genes, what are the mutations you should do to control your system. So this relies a lot on associative programming, but we have, on top of it, we have kind of a modeling language, modeling platform where we can specify all these domain-specific properties. So we have keywords so it's in Python, where we have some kind of syntax to express reachability, fixed point properties. So we can, like that, model our synthesis problem in Python, and then it will compile in ASP and use this generic solver to do optimization and find solutions and so on. So in our case, yeah, so if you want to try ASP someday, if you're curious, you can use, I really advise using this Klingo solver. So it's very efficient and very nice language to use. So we apply it on different biological systems. So I won't go into detail, but just to show you how it works, the general workflow of this model synthesis, how we go from data to a model that can reproduce the data. So we have some data, we observe the cell in different states, we transform it into Boolean properties. We have also prior knowledge on what is possible or not in biology, and then we apply this synthesis problem. That what we do is we do it in several stages of optimization. So, as I said, in cells you have thousands of genes, but typically in biological process we observe not all of them are important. So, okay, we find Boolean network with thousands of genes, but you can optimize and find the smallest, the simplest model that can reproduce the process. And so, using this combinatorial optimization, you can automatically select from the data and the property what are really the important parts of your system to be able to reproduce what you observe. So we do this gene selection, this subset, this minimization of model using this combinatorial optimization within ISP. Yeah, so to conclude on this, one of the remaining challenges is, okay, so now we are able to automatically build models that have the property we want. So it's, and we can do it at a realistic scale, but it's only formal things, so we have no way to assess if the model is predictive or not. So if I give it new data, is it likely to be able to predict this new data? This is something in machine learning they do a lot. Right? They spend some time doing only that. So you have training, testing data, and then you have optimization procedure to maximize your predictive power. We don't have that in this formal synthesis approach. So we are working on how to try to mimic what is done in machine learning in this formal synthesis approach. And we still need a lot of new theorems on abstractions, QBF solving, etc. Model counting. Model counting is very important for us. We have no idea how to do it. Because systems are very large, so if nothing works. So we still need a lot of theoretical tools. So yeah, so to conclude, so as you've seen, so we had a quite broad challenge, and to tackle this, we had to use many different fields of computer science. So we use theory of dynamical systems, abstract interpretations, computational complexity, logic programming. We also use biology and data science more and more in the group. So we really need to interact with a lot of communities, and this is very, very interesting. Super. Yeah, we learn a lot of things every day, thanks to all these people. So that's yeah. So that's all of what I wanted to say. So just so currently in my group, so we have. I'd like to especially thank Gustavo, Theo, Celia, Deborah, and Snails, who did several work related to what I've shown you, and different colleagues in France and Europe and South Korea. So thank you very much.
Okay, on time. But if you have some questions. Any question?
Actually, I have one question about the transformation between the states. So in the earlier talk of your presentation, that you said that you can find a way that you can transform the differentiated cell from this cell to this cell by like assigning 1 to second bit. And in that case, one more important thing is that, is it really possible? Right? So if we can set F2 as 1 in the model, but in real life we cannot do it.
Sure.
And then it does not really matter, right? So do you consider that kind of things in the modeling, or is it purely just computation?
No, sure. Okay, so that's several things. So in practice, you do not find many solutions. So when you find a solution, for instance, set F2 to 1, already from a biological point of view, it tells you that this gene is very important. Maybe you're not able to deal with it, but at least you gain the knowledge that this is a sensitive point for your system. I said here, these are genes, so often we are not able to interact directly with the gene, but you may have drugs which indirectly, through signaling, will tickle this gene the way you want. And so we have databases of drugs, and we can then try how to act on it. And then, yeah, we have some genes we know, and but we cannot deal with that, so we can exclude them. And since like prior knowledge we can take advantage of to just say, okay, we won't be able to teach you.
Based on actually, I had a similar question, but I asked the professor in biology. They said it's really possible if we try to put some chemical or something, we can permanently or enforce to certainly to be 0 or 1.
So you have heard about this CRISPR technology, notably, so very famous. So it's good to cut genes, but to force some genes to be active, it's a bit more complex because you need to make sure all the material is here. So yeah, depending on the constant you put and which you act, can be complex.
In the conclusion and the limitations you mentioned is about the gene reduction. You should be doing it somehow, even though there is no textbook for it. Like all the cells in our body have all the genes. However, we cannot put all the interactions into one model. How is it? How is it? Is there any as-is process for the node selection before putting it into the model?
Yeah. So the thing is, some genes, so you may have no observation of them, but they may play an important role because they have an influence on. So you cannot really just from the data do the node selection. Here, this is really justified by the dynamical model. So how we do it is that we first find a Boolean network with a function for the 1,000 genes, and then we have an optimization objective which says try to plot as much gene as possible to a constant value. And the idea is that if a gene has a constant value in all the process, maybe it doesn't play a role, so you can then remove it. So we first optimize and try to plot as much a gene as in a dummy constant function, and then we move those genes. And those who stay are really important because they have dynamics, which is necessary to reproduce your data. So this is how we identify. But this is really justified by the dynamical model, not by the data.
So like the dynamical model and iterative process of selecting and deselecting genes.
So this is in the synthesis process. So in the ASP, we have like an optimization objective to say, put as much as gene to constant function when possible. And this is how we identify non-important genes by combinatorial optimization.
Yeah, I don't want to exceed too much the time. So yeah, thanks to the speaker again. So this is end of the first seminar. 한 걸로 할게요 이제. 너무 신기해가지고 좀 더 다음번에는 조금 더 프렌들리한 분위기에서 했으면 좋겠습니다. 안 그러면 안 올 것 같아. 다음번에도 또 즐거운